Now, next, we are going to discuss in this lecture about the physical factors which affect the growth of any given organism. So, I repeat again, under the point of requirements for growth, there are two types: nutrition requirements and physical or environmental factors which affect the growth of bacteria. So, what are these physical factors? Here we are just enlisting all those factors which are required for growth of bacteria. These are pH of the media, temperature of the environment, then level of oxygen in the environment, then level of moisture, then hydrostatic pressure, osmotic pressure, and radiation. So we are going to discuss one by one how this each point is going to affect the growth of any given organism. So out of these seven factors, first four factors are very vital and very important, as well as long as culturing of organism in the laboratory is concerned. So let's have a look at these factors one by one. Let's start with pH. Okay, so before that, as we have already discussed, we need to understand that bacteria, among all the prokaryotes, bacteria are the organisms which can survive and thrive in any kind of environment. So we must remember that on this land, almost 70% of the land of the earth is filled with water, 73%. And in that water, a majority of the portion is salt water. There is hardly any content of water which is termed as fresh water, which is directly available to us. But the majority of the portion is available in the form of salt water. So it is just hardly a very small fraction which is occupied by warm-blooded or land-dwelling organisms like us. So other major portion of this planet is occupied by different species of bacteria. So bacteria can survive in very extreme environments. It can grow and thrive in salt water, in fresh water, in very deep sea environment, in hot springs or in hot lakes, or in polar regions, and in under icy lands, or under like even below 0 degrees Celsius in polar lakes. So there are all kind of environments in which bacteria can be found, surviving and growing very well. So these are this picture depicts all kind of environments in which the bacterial growth is possible. So sea ice, polar region, the deep sea and brine areas, deep sea hydrothermal vents, or volcanic regions, or hyperacidic lakes, deserts and arid environments, and drainage, or nuclear waste sites. There is no region or no area on the earth in which is like not supporting growth of any living species. So bacteria is one organism which is found in all possible environmental factors. So this is one example of an extremophile. By extremophile, I mean an organism which can survive and live in the extreme environmental conditions. So Conan the bacterium, Deinococcus radiodurans, is one organism which has been found to survive the blast of gamma radiation, which is 3,000 times lethal dose than for humans. So this tiny organism, tiny microscopic organism, which is like not even visible to naked eyes, it is that small, and this survived which the dose of radiation 3,000 times lethal than humans can survive. So in 1999, US Department of Energy tried to research on this bacteria in the hope to decode its genomic structure, in the hope that there is something genetically different in this organism which is helping it survive such a such a major attack of radiation, and if they could decode its genetic structure, it can be used to produce certain kind of species by genetic engineering, which can help us in degradation of the contaminated nuclear sites, or contaminated or nuclear waste degradation disposal. But it was found that there was nothing special in the genetic material of Deinococcus radiodurans. So its DNA was just as susceptible as E. coli or any other bacteria's DNA was to radiation. But the speciality for this organism was it had a set of antioxidants or a set of enzymes it was producing, which was protecting its proteins from radiation damage. So as soon as the DNA or other proteins were getting damaged by radiation attack, that antioxidant or those enzyme sets were immediately repairing those DNAs, those damaged DNAs. And that is how this organism managed to survive in the radiation attack. So research is still going on such kind of organisms, so as these will help us in constructive methodologies to maintain or conserve our environment back to normal. Now, another example is this species which is known as water bear. So this water bear or tardigrade is known as multi-extremophile. So this is one like amazing species. Um, it has ability to survive very harsh environments, such as extreme heat, extreme cold. It can survive desiccation. It can survive in environment lacking oxygen, and it can also survive radiation. So the single bacterial species can survive all these extreme conditions. They have also carried out experiments in which they found out that this tiny animal also survived a 10-day trip into the space. So uh, they it gave them hope that it can be a certain type of creature which can also survive on Mars. So much research has been carried out on this bacterial species, but it was found out that this species kind of goes into hibernation, or it puts itself into a form of non-reproductive suspended animation. Okay, as long as it is facing any harsh condition. So in during that hibernation condition, it just stops, its metabolic processes stop within the body, or everything is stopped, but it doesn't die. So once the favorable conditions return, it comes out of that hibernation and carries out with its normal life. So there are certain species which have been shown hope to survive, or they have the ability to survive in extreme conditions, by which they give hope that those species can be surviving, can also survive on the Mars, and they are trying to carry out the research by sending such a species sample on the Mars, and trying to collect it back in the hope to find out more information about the environment on the Mars. So these are few examples of miraculous organisms which can survive in extreme conditions. So now let's come back to the factors which affect the growth of normal bacteria, or the factors which you should consider while culturing the organisms in the laboratory. So first factor is pH. So all the organisms or all the species of bacteria have optimum pH, or the pH at which they grow the best. So, as per their tolerance to acidity or alkalinity, or we can say low pH values and high pH value, they are categorized as acidophiles, neutrophiles, and alkaliphiles. So, as these names suggest, acidophiles are the organisms which are known to be acid loving, or they grow best at low pH ranges, pH range of 0.1 to 5.4. So these organisms can survive, or more probably thrive at very low pH ranges. If you put these organisms at neutral or basic or alkaline pH, they will die. So acidophiles only survive at very low pH. Neutrophiles are organisms which exist in the neutral pH range, or they can grow their best in the pH range of 5.4 to 8. So most of the bacteria that cause diseases in humans are neutrophiles, are the organisms which can grow in this neutral pH range, or this is the pH range within the human body, inside the human body. So that is why neutrophiles find it favorable inside the human body, and they can cause infections. Now, third category is alkaliphiles. Alkaliphiles, as the name suggests, are base loving or alkali loving. They grow their best at alkaline pH, or the pH ranging from 7 to 11.5. So, as we discussed already, these bacteria have these optimum pH ranges. So, these are the pH ranges at which they will grow their best. If you put acidophile at the pH of 10, then acidophile will die. It cannot survive outside its pH range. If you put alkalifile in the pH range of 2 to 3, it will die. Also, same rule applies to neutrophiles. If you put them outside their pH range in either acidic or